In this video, we'll go over step 6 of 6.
Let's open up the KL14F file first. To find the first SNP in TAS2R, we're going to look for the reference sequence AGAGGCAG. We can see our reference sequence right there. And our SNP of interest is the next downstream base pair. So here we can see that there's actually two overlapping colored line traces. And this implies that we have a heterozygous locus. In other words, we have an individual that has received a C from one parent and a G from the other at this first SNP. So to find the second and the third SNPs that are within TAS2R38, we need to look at the reverse sequence read because the PCR product for TAS2R38 is longer than the sequence read length. So first we're going to open the reverse read file for the same sample called KL14R. And the first most important thing we want to do is reverse the sequence read to its reverse complement. Now that we've reformatted TAS2R38, we can now locate the reference sequences for SNPs 2 and 3. To find SNP 2, search for ATCCTGTG. And zooming into this region, we can see that the next downstream base from our reference sequence has two overlapping colored lines as well. So in this case, we can see that this individual has received a T from one parent and a C from the other at SNP number 2. Finally, let's find SNP number 3 for TAS2R38. Search for the reference sequence TGCAGCC in the reverse complement sequence for the reverse strand. Let's zoom into this region and we can see the next downstream base has two overlapping colored traces. And in this case, we're looking at an A and a G that are overlapping. So our example genotype for TAS2R38 is CG heterozygous at SNP number 1, TC heterozygous at SNP number 2, and GA heterozygous at SNP number 3.